Hi everyone, and welcome back to my channel. So in this video, I'm going to talk about pedigrees and how we can use those to trait the inheritance of traits, as well as those that can lead to human disease. So at some point in your life, you may have seen a chart like the one shown on the left here. And this is what we call a pedigree chart. And what's neat about these is they allow us to track inherited traits across generations. So, one of the things I've talked about in another video is how we can use genetic crosses to look for things like modes of inheritance and that kind of stuff. Well, that's great if you have the ability to manipulate some type of genetic model organism like C. elegans or Drosophila or even mice and rats. It doesn't work well when you're trying to track traits in species where that type of experimentation is either very challenging or completely unacceptable. So, for example, humans, or this is often used to track dog breeds and that kind of stuff. So, you've probably seen something like that that looks like this. They can be in black and white, they can be in colors. Usually, there's a key somewhere to tell you what each of the different shapes and colors or the lack of color actually means. So you can see in this example that I'm giving you, and in most cases, squares are going to represent males, circles are going to represent females. You may also see other color or other shapes used in some cases. So, for example, triangles might be used when you have an ancestor of unknown sex. We don't know whether they're male or they're female. And then what you're seeing here in the key, for example, in this particular one, it's not always this way, but in this one, blue coloration represents somebody who has the wild type, the wild type alleles for this. Then you have half filled in circles that are half blue and half red to represent individuals who are heterozygotes, and then you would have all red ones for those who are homozygous for a given trait. So the diagrams are used; they'll show the phenotypes and or the genotypes for particular organisms and its ancestors. And like I said, this is very commonly used for either basic research purposes. It can be used for tracking the inheritance of certain alleles in humans and other animals. And these are often used for genetic counseling. So people might be considering having a baby, but they know that there's a history of some inherited diseases in one or both sides of the family. And they want to find out what is the chance that our children could potentially have one of these diseases. When you're constructing a pedigree chart, you should be including all information that is available to you. And this is important, right? The more information you have, the better the estimates you can make in terms of the likelihood that a trait is either being carried by an individual or could be passed on to a subsequent generation. But this can be challenging for a lot of reasons. First off, we're looking at 3 generations right here. So let's just say that this is a child, right? This is somebody who's a child, maybe 10 years old. Now we're looking at the parental generation. These people are likely going to be in their 30s, maybe even in their 40s at this point. And then we have the generation prior to that, so the grandparents, and now we're looking at people that are well into their 70s. So we're relying on, a lot of times, we're relying on the memories of family members, right? So we might not even know who this aunt and uncle are, but we're hoping that grandma and grandpa might remember whether their brother and or brother-in-law, sister-in-law has some sort of illness or might have some sort of disease or anything like that. And once you get past the living generations, you start getting up in here into the great grandparents and beyond. Now we're relying on memories that are decades, even centuries old at this point. So it can be very, very hard. We're also oftentimes relying on old clinical diagnoses. I mean, the fact that we're in the year 2025 right now, as I record this, means that, even going back into the grandparents' generations, there are pretty adequate medical records for those individuals throughout the majority of their lifetime. But once you get past that, they become less reliable. Also, clinical diagnoses have changed over the years. We've gotten a lot better at diagnosing certain diseases, understanding what they are. Some people may have had a specific disease, but we didn't know what that disease was at the time, or it was called something different, or it was just sort of ignored. And then we go, well, I don't know, right? Like we're relying on old clinical diagnoses. Some other confounding variables include incomplete penetrance. So there are people in the cases of some diseases, in the cases of some traits, that people can have the allele, whether it's heterozygous, homozygous, but they may not express the phenotype. So not every single one of these automatically, I mean, you have the allele, you get the disease. It's not always like that. There are some cases where the penetration of those alleles isn't 100%, meaning you might have somebody who actually was heterozygous or homozygous for something, but since they didn't show the phenotype, nobody knew. Also, it can happen in an age dependent manner, right? So let's say that this is something, I can give you an example, something like Huntington's doesn't usually show up until the 40s or the 50s. When I mean somebody gets to their 40s and their 50s, it can happen earlier, but often it isn't until middle age where you start to see signs of this. Well, what if somebody died in their teens or their 20s of something completely unrelated? Car accident, they died in war, there was, they got cancer, something else, they died young. We would never know if they were going to develop that disease phenotype, so we might not have that information. And there's also something called variable expressivity, which means the phenotype can vary with respect to how strong the phenotype is, right? You might have somebody that has the disease alleles and they suffer from the disease, but it's so minor that they might not know they have it, or they just kind of go, that's kind of how it is. Whereas other people with the disease alleles have very profound diseases associated with it. So when you factor all this in, pedigrees aren't always perfect. They're not always the most reliable, but sometimes it's the best thing we have available to us. Now, typically a pedigree chart is going to begin with someone we call the proband. This is typically the individual who brings the family to the attention of a geneticist. So it's like why we're here. Why are we making this? It's the person who's asking us to make a pedigree chart. And again, this could be somebody who's thinking about having kids, but there's a history of some genetic diseases or genetic conditions that has been passed down through their family, and they're wondering whether or not they're at risk of passing this on to any potential children they may have. There's then we start to build that based off of the information that we have available, right? Hopefully we can get a hold of medical records, but it may also involve that proband going back and asking their parents, asking their grandparents, aunts and uncles, hey, did so-and-so have this particular illness? Did so-and-so have this particular disease? Did you ever notice this with somebody? And you collect as much information as you can. Now, as I mentioned, when we construct these things, colors, shapes, they're all very important. Like I said, typically males are represented as squares. Females tend to be represented as circles. Triangles can be used if the sex is unknown. I don't mean to say, some people will throw diamonds in there for sex is unknown. Just make sure you're paying attention to the key. When you make one of when you make a pedigree chart, there should be some sort of key to help people understand. We also use partial shading when somebody's a carrier, so a heterozygote, but because of the different types of dominance we see with respect to these alleles, we might not know that somebody's a carrier. So we might have them listed as unaffected, but not know that they're a carrier for it because we just don't know, because perhaps the disease allele is homozygous recessive. So sometimes we don't have all the information. So what I'm going to do now is we're going to go through a series of pedigree charts that show different types of inheritance. And again, this is a way that we can sort of learn about what mode of inheritance might be being experienced with respect to a particular trait or disease or whatever. Again, not always perfect, right? So let's start with a very simple one, autosomal dominant. So if you recall from our modes of inheritance, when we have complete dominance, okay, that means a single copy of the dominant allele is enough to dictate the phenotype. Okay. So if something is inherited in an autosomal dominant fashion, every person with that allele will demonstrate that phenotype, assuming complete penetrance, right? There is always that exception that sometimes people don't show the phenotype, even if they have the allele. When we see something that exhibits autosomal dominant inheritance, we also see that typically every generation will have at least one affected individual. Why? Because as long as one of the parents has an allele, there's a probability that it's a good probability that will get passed on to the subsequent generation. And since even a single copy of this autosomal dominant trait is enough to give the phenotype. All right. So for example, here we have an affected male, a wild type female. They have children, 1, 2, 3, 4. So they have 1, 2 daughters, they have 1, 2, 3 daughters, and they also have 1, 2 sons. one of the two sons is affected. Two out of the three daughters are affected as a result. And then we can see, because these children pass it on to what we can refer to as the grandchildren, we see it also showing up in the third generation as well. So the first parental generation, if we're talking crosses, this would be the F1, although we typically refer to it as the second generation because we didn't cross these people, and people don't like to refer to having kids as having a genetic cross. And then we get to the third generation, and we can see that there are individuals at every level who are affected, but not everyone. There are unaffected individuals that have either married in, or I should say, we don't have to assume marriage, but we're included in the production of offspring here, and it gets passed down from generation to generation. Autosomal dominant diseases are typically rare in populations because the affected individuals and affected individuals tend to be heterozygous. It's rare because if you're going to have two individuals that are affected, to get a homozygous dominant or a homozygous individual, both parents would have to have that particular syndrome, disease, whatever, to pass it down. So we often see a lot of heterozygosity when it comes to these. So here are a few examples. Achondroplasia, which is a form of dwarfism. If you remember Wee Man from the show Jackass, he is someone who has achondroplastic dwarfism. You can see him here, and you can see how this gets passed down. It's an autosomal trait, which means it's not on one of the sex chromosomes. You have one affected parent. If you have one affected and one unaffected parent, it's basically 50-50 whether it gets passed on to the subsequent generation, because there's a 50% chance that a child would inherit the affected allele from their affected parent. Polydactyly, which is a trait of having more than 5 digits on any one of your appendages, is also something that is passed down in an autosomal dominant fashion. What about autosomal recessive? So for an autosomal recessive trait to appear, both parents would have to carry at least one copy of the disease allele. All right. So you can see in this pedigree chart, one of the things that differs. So first off, it's a different chart, right? We have different colorations. We're using light blue and red. We've got a different breakdown in terms of how we're assigning these things. They now in this case, they're referring to as normal as opposed to wild type. Just make sure you're checking your key. I'm just reminding you of that. So here we have two parents in the first generation who are heterozygotes. Okay. It's an autosomal recessive disorder. They may not even know that they are carriers of this particular disease. And we would typically refer to them as carriers because they're not going to be affected. They're not going to have the phenotype. But then these two get together. They have 5 children. You can see that they have 1, 2, 3 girls, and they have 1, 2 boys. All right. Now, one of the things you're going to see right here, this individual is a carrier, but unaffected. Carrier but unaffected, carrier but unaffected, not a carrier. And one of these 5 children, 1 out of the 5, is homozygous for this particular allele. It happens to be a male. One of their sons is going to now show this particular disease phenotype. All right. But the other thing to note is 4 of the 5 children possess that allele and then can pass it down to subsequent generations. So let's look at one case example here. We have an individual who is a carrier. They go on. It is a daughter. The daughter goes on and has children with another man who is not even a carrier, just a normal man. And they have 1, 2 children. Both of them are males. One of them is a carrier. The other one is not. Over here, we have someone who actually has the disease. They are a homozygous male. They have 1, 2, 3 children, 2 sons and a daughter. All 3 of them turn out to be carriers. So even though this individual is homozygous for this, he can only produce carrier offspring because the individual he is having children with is not a carrier at all. Note that all of the children will have to be carriers though, right? Because this individual is affected. All right. And you can see that as we go down to the 4th generation, we are now getting into several more carriers. One thing to note though, nobody in this 3rd generation is affected. They might be carriers. Nobody in the 4th generation is affected. They might be carriers. All right. So this is the thing that we have to understand. We see these autosomal recessive traits. We're going to see multiple generations where we don't have affected individuals, but we see carriers. All right. And again, We might not know that they're carriers because if there is no outward appearance for a heterozygote in this case, which there's not, you might not know. But for example, all three of these individuals should have known that they're carriers because their father has that particular disease phenotype, meaning he could only pass on this particular trait. Some examples of autosomal recessive diseases. Tay-Sachs, which is a lysosomal storage disorder that is universally fatal at a very, very young age. This is inherited in an autosomal recessive fashion. Sickle cell anemia is inherited in an autosomal recessive fashion. Phenylketonuria is a disease where people cannot process the amino acid phenylalanine, and they have to be very careful about how much of it they consume because it can make them ill. You may have seen this on almost any can of diet soda. You're going to say, warning, contains phenylalanine. Why? Because the fake sugar they put in there, aspartame, has phenylalanine associated with it. And as a result, people who have phenylketonuria need to be careful with this because it can lead to an overabundance of this. Cystic fibrosis, another example of an autosomal recessive disease that's inherited through this modality. So what if it's not an autosomal trait? What if it's an X-linked trait? So this would be a sex-linked disease that's associated. So if it is X-linked dominant, all right, X-linked dominant traits tend to impact females more than males. So over here, if we look at this chart, we are going to have square males, circular females. I just don't have the legend associated with it right now, just in the interest of space. But again, this purple coloration is going to represent affected individuals. Half colorations represent carriers in the case of the recessive. So let's compare X-linked dominant versus X-linked recessive. So again, this is going to be inherited in a sex-linked manner. It's going to depend on which parent has it in some cases and what the sex of the child is, whether or not they're going to show this. Now, X-linked dominant traits tend to impact females more than males, with the main reason being they have 2 copies of the X chromosome. So they can get it from either parent and show that particular phenotype, all right? The other thing to note, in this case, we have an affected father. All of his daughters are going to have this particular trait. Why? Because this father only has 1 copy of the X chromosome that he inherited from his mother, who passed this on to him. And because he is automatically going to have to give that X chromosome to any of his daughters, all 3 of his daughters are affected, but none of the sons are, because the mom only has wild type X chromosomes and dad is providing his, what we would presume, healthy Y chromosome to them. So here you see it's the females. In the 3rd generation, still the females. We do have a few affected males getting involved here because these females are having kids, which means they will be passing their X chromosome, one of their X chromosomes, on to their sons. But we don't know which one. So here's a great example. You have the father passes it on to his children, all 3 of his daughters. Note that 2 of them didn't have offspring here, right? 2 of them didn't. But then in this generation, we see that the granddaughter does get the X-linked trait from her mother. Her sister did not, though. Mom gave it to one, but not both children. And then down here, she passes it on to her son, right? Because she has to provide an X to her 2 sons. One of them got the wild type X and one of them got the disease X. He now has it, but note that his son doesn't. Why? Because he can't pass that X on to his son. If he had a daughter, though, then she certainly would have had it. So you can see that in the inheritance pattern. All right. When we look at X-linked recessive traits, it's different. It's typically the males who are going to be affected. Why? Because when mom passes it on, it's possible that the daughters, the mom can pass it on. The sons are going to have it, right? Because they had to get an X chromosome from their mom. All right. So they're likely to have it. The daughters are going to be carriers. So mom is affected. So because mom's affected, she has 2 copies of this, right? She has to have 2 copies of it to be affected. It's a recessive trait. So mom is affected. Mom has the disease, the syndrome, the whatever, the trait, the whatever. When they have sons, mom's going to give it to all of her sons. Has to, because mom's giving one of her 2 mutant copies, disease copies to the son, dad can only provide a Y. There's nothing to balance that out, right? Daughters are carriers. So X-linked recessive in this first generation, we have an affected mom. The children are either going to be carriers or they're going to have the disease in the case of the son. Daughters carriers, sons affected, right? And then we look at the next generation. This particular carrier mom gave it to one, but not both of her sons. All right. This particular carrier did not choose to have children. Over here, we have an affected dad making both of his daughters carriers. Moving on, this particular carrier then had children with an affected male, and they pass it down. Now, again, when I say diseases, it could be syndromes. Let's look at some examples of these two traits. So when we look at X-linked dominant inheritance, this does a very good job of showing with the X and Y chromosomes how this inheritance pattern works. So if we look at X-linked dominant, if we have an affected mother, she will pass it on in about a 50-50 dosage to her son and her daughter. Why? She can either pass on the mutant version or the normal version of that gene. So 50-50. If it's an affected father, all of the daughters get it, the sons don't. That's a very, very dead giveaway that this is an X-linked dominant trait because, again, we talked about reciprocal crosses in one of my previous videos about how we can use reciprocal crosses to uncover sex-linked traits. This really, if you think about it, is nothing more than a reciprocal cross. If mom has an autosomal dominant, an X-linked dominant trait, then there's a 50-50 chance that her offspring get it, whether it's a son or a daughter. The other side of this, if dad has it, his daughters both get it, his sons don't, right? So that's a good clue that you're looking at an X-linked dominant trait. When we look at it in terms of X-linked recessive, well, if it's X-linked recessive, dads are going to be affected if they have it, right? Because they're hemizygous. They have that one X chromosome. Whatever this X chromosome says, that's what he gets. So in this case, dad's affected. His daughters are automatically carriers because that's the only X he can pass on. His sons are going to be unaffected because he's giving the Y. On the other hand, if mom is a carrier, she can have unaffected daughters, unaffected sons, carrier daughters, and affected sons. So the sons are 50-50. Why? Because they're getting their dad's Y, and then it's a 50-50 coin flip whether they get the mutated gene from mom. And if they get the mutated gene, they're affected. If they don't get the mutated gene, they are unaffected. Daughters, on the other hand, if they get the gene, they're a carrier. If they don't get mom's gene, they're unaffected because they are balanced out by dad's healthy X chromosome. So you can see that if you look between these two different slides, here's what it looks like in terms of the X being inherited. Here's what it looks like when we build the pedigree. So it gives you a very clear idea of how we can use a pedigree to go, oh, this is what's happening. This is the mode of inheritance. So here are some examples of X-linked dominant either diseases or conditions that are inherited in an X-linked dominant fashion. Vitamin D resistant rickets. So rickets is a disease typically caused by either a consumption of a low amount of calcium in the diet or a low amount of vitamin D, since vitamin D is required for calcium uptake. But in the cases of vitamin D resistant rickets, people develop rickets at a young age, even though they're getting the proper amounts of it in their diet, simply because the vitamin D isn't effective in getting that calcium imported the way it needs to be. Alport syndrome, incontinentia pigmenti, which you see here, is another inherited condition that is inherited in an X-linked dominant trait, or an X-linked dominant fashion, sorry. And then we have X-linked recessive conditions and diseases. So, for example, red-green color blindness. So hopefully the top half of this picture looks different than the bottom half. If not, you're red-green colorblind. You probably know that at this point, but if you didn't know that, you should probably know that now. For those of you who aren't red-green colorblind like me, you can see the red apples and the green apples, but down here they look very similar. Why? Because of the red-green colorblindness. Hemophilia A and B are both inherited in an X-linked recessive fashion. If you want a very, very interesting story about how this was passed down through generations, go look up the European royal families just prior to World War I. And you'll see how Queen Victoria then passed this down to basically every royal family in Europe. There's a really neat story wrapped up in all this about Rasputin and the Russian Tsar and Tsarina and how that led to the Russian Revolution. Really interesting story if you want to know how an inherited disease can impact global politics. That's an example. Duchenne muscular dystrophy, X-linked agammaglobulinemia, which is an inherited condition where people have an inability to produce antibodies of certain types here. So, by the way, I mentioned the hemophilia A and B. Here's Queen Victoria, and you can see how she passed it all down. Eventually, to one of her, so this would be her children, grandchildren, great-grandchildren, Alexei, who was the son of Tsar Nicholas, who was married to one of the granddaughters of Queen Victoria, Alex, and passed this on. And then Rasputin got involved because he thought he would be able to treat Alexei, and that led to a whole bunch of unhappiness within the upper echelons of Russian society during World War I and contributed to the Russian Revolution. So here's what that looks like here. Very interesting story. Seriously, go look it up. I don't have the time to talk about it here, but worth a look if you're at all interested. And then we also have a couple other types of inheritance that can be detected this way. Y-linked. So again, I talked about this in another video. It's called hairy ear rim syndrome. And it's very easy to spot these because only males are impacted. If you look at a pedigree and the only people who have these diseases are males, it's probably Y-linked. But it's also very rare because the Y chromosome has very few genes on it to begin with. So it's rare to find these conditions. There are also mitochondrial diseases that can be passed down. Interesting about these, just recall that mitochondrial DNA always comes from the mom, so dad can never pass these on. So if dad has it, he can't pass it on to the next generation because all the mitochondrial DNA in the children, male or female, are going to have moms of mitochondrial DNA. They are also very rare. Now, mitochondrial diseases themselves are not rare, but most are sporadic in nature. Very few of them, but here are some examples: Carnes-Sayre syndrome, MELAS, and then also MERF. And you can see a woman who's been impacted by one of these diseases here. These can be inherited. They can all, and I would notice that all three of these diseases can be inherited or sporadic. So, it depends on what disease allele you have, whether it's inherited or not. Okay. So one of the reasons people have pedigrees made, the proband, if you will, is because people are often curious, what is the probability of passing a particular trait on to their offspring, right? So they'll go to genetic counselors to figure this information out, all right? So how can we actually calculate a realistic possibility of this, right? Now, again, I'm going to give an example here. I'll work your way through it, show how we can use the product, some rules, that kind of stuff to do this. But what I want to be clear about with this is we're assuming that we have all of the information that we can have. And that's not always the case. So we can't always give a 100% estimate. And you'll kind of see this as we go through, that we're relying on some assumptions that may not necessarily be true, but it gives us the best, most likely possible answer we can get. So we're going to use this as an example of a disease that has autosomal recessive inheritance. All right. And the question that we're asking is, what is the probability that individual number 14 will be affected by this disease? So let's look at the scenario that we have. Let's assume that this woman, number 12, and this man, number 13, they're planning to have kids. Now, notice that we had number 14. Notice number 14 is a diamond. That means we don't know the gender of this child. We don't know the sex of this child. We don't know it because they don't exist yet. So we're trying to calculate if this woman and this man have a child, what is the probability that this child will be impacted by this particular disease? Okay. Now, we can assume, here's what we're going to assume. We're going back to 12 and 13's grandparents, right? Here are their parents. Here are their grandparents. We are going to assume that numbers 1 and 2, grandparent set 1 and 2, grandparents set 1, and 3 and 4, the other side of the family, grandparents, are at least carriers. So why can we make this assumption? Well, here's why. First off, none of these individuals has the disease. They're not affected by this. Whatever it is, they're unaffected individuals. But they've each had at least one child that has been impacted and is affected. And the only way that can happen is for them both to be carriers, because this is an autosomal recessive trait. Okay. So we can look down here at number 6 and number 9 and assume that there is at least a chance that 6 and 9, the father of 12, the mother of 13, okay, have the potential to be carriers. But what's the probability of this? Here's how we would look at this. Numbers 6 and 9 have equal probabilities. So we can take this mathematically. Here's why. The potential genotypes for both of these are to be homozygous wild type, heterozygotes, or they could be homozygous for the affected allele, for the trait. But we know that they're not homozygous for the trait. We can rule that that possibility out. Why can we do that? Because they're not affected. So we can rule homozygous for the disease, we're going to call it a disease, homozygous for the disease allele out. Can't be, because they would be affected. So that means there's really only 2 potential outcomes. Homozygous normal or heterozygous, a carrier. All right. So instead of, because and usually the genotypic ratio using Mendel would tell you that it's 1 to 2 to 1 for this. All right. 25% chance they're homozygous wild type, 50% chance that they are hets, carriers, or 25% for being homozygous for the disease allele, which would mean they're affected. But they can't be this because they're unaffected. Therefore, there is a 1 plus 2, a 2 in 3 chance, 2 out of 3 chance that they are heterozygotes. Both of them, right? Because we can rule out this. They're not that. We know they're not that. But there's still a 2/3 chance that they're a carrier. So how do we look at that in terms of the next generation? Each of these parents, all right, of 12 and 13 has a 2 in 3 chance of being a carrier. But we also have to factor in that they are not the sole provider of genetic information for the next generation, because they have to mate, reproduce with somebody to make 12 and 13. And we are going to make the assumption that 7, the woman who has children with 6 and produces 12, and 8, the man who produces children with 9, 13, are not a disease carrier, and we know they're not affected. Why are we making that assumption? Well, let's just assume that there's no history of this particular disease in 7's family or in 8's family, which would mean there's not really a chance that they're a carrier, and we know that they're not affected. So, using this probability, using this math, there is actually only half of a 2/3 chance that number 12 and number 13 are a carrier. Why? Because there's a 2/3 chance that they're a carrier in the first place. Only a 2/3 chance, right? Because there's still that 1/3 of a chance that they're homozygous for the wild type allele, that they don't even carry the trait. But we also have to take that 2/3 of a chance and cut it in half. Why do we have to do that? Well, simply put, there's only a 50-50 chance that if they carry the disease allele, it makes it into 12 or it makes it into 13, right? That's what Mendel's law of segregation would tell you. They could either pass on their disease chromosome or their normal chromosome, their disease chromosome or their normal chromosome, because the other copy of that chromosome is coming from 7 or 8, right? To make number 12 and to make number 13, respectively. So what that means then, using our product rule, 2/3 times 1/2 equals 1/3. All right, from 6. There's a 0 chance that 7 is going to provide it, which means number 12 is a 1 in 3 chance of carrying the disease allele. Okay? The same math applies for the mating between 8 and 9, the pairing of 8 and 9, to produce number 13. Number 13 also has a 1 in 3 chance of being a carrier. That's it, a 1 in 3 chance. So hopefully you followed along with that math. But again, we're just using the product rule. Okay, so then we now know that the probability of 12 and 13 being heterozygotes, being carriers, is about 1 in 3. All right? So the next thing we have to ask ourselves is, what is the probability that 14 will be affected? Right? Because that's the question we've been asking all along. What's the chance that hypothetical child number 14 will be affected, will have the disease? So again, we go back to the product rule. Each parent has a 1/3 chance of being a carrier. All right. So in order for this child to be affected, both parents would have to be a carrier. So the first math we have to do is, what is the probability that both 12 and 13 are even both carriers in the first place? This is our AND rule. This is, again, the product rule, because we need 12 and 13 to be carriers to even have a chance that 14 will be affected by the disease. Well, 1/3 times 1/3 tells you that there is a 1 in 9 chance. So mathematically speaking, there's only a 1 in 9 chance that both 12 and 13 are even carriers at the same time. Okay? So we're already like, okay, well, 1 in 9, that's not terrible. But also, in order for 14 to be affected, they would have to both be carriers and pass on that 12. And number 14 would also have to inherit both of the disease-causing alleles. So what's the odds of that? Well, again, we go back to Mendel's crosses, right? We go back. There is a 1 to 2 to 1 ratio in any case where parents are both heterozygous at a given allele, which means there's only a 1 in 4 chance of 14 also being homozygous for the disease allele in the case of both parents being carriers. So we have to factor that in as well. So now the entire equation becomes the 1 in 3 chance that mom's a carrier times the 1 in 3 chance that dad's a carrier times the 1 in 4 chance that if they were both carriers, that 14 would inherit both of the disease-causing alleles. And 1/3 times 1/3 times 1/4 gives you a probability of 1/36. So with the knowledge that we have, with the information provided, these two know that they have about a 1 in 36 chance of passing this on to their child. And that's just from doing the pedigree. Now that we have more information, we have more access to things like genetic tools and things like that, we may actually be able to look at them genetically and be like, hey, you're a carrier, but you're not. There's a 0% chance that your kid gets this. But this gets us started on the process. This gets us to the understanding of, yeah, you got about a 1 in 36 chance of passing it on. There's only a 1 in 3 chance that either of you are even carriers to begin with. So maybe at that point they save the money and go, well, we don't need to worry so much about doing all these really expensive tests to test for this, because the chance of my child is of our child inheriting this is very low. It's comfortably low by our standards. So this is a great example of how we can use pedigrees to calculate the probability of passing on a certain trait to subsequent generations, which is really neat. And this is why people have done this for a very long time, to try to figure out this stuff. Again, with the advent of newer genetic tools and genetic testing and stuff like that, these may become less and less relevant, but right now, still very helpful. And also just a very interesting way of studying the inheritance of certain traits. And as I said before, even without this, using pedigrees to figure out the modes of inheritance is a very, very useful thing, especially when we discover a new disease or a new inherited condition or a new allele that's appearing in a certain population. This is a great way of figuring out, like, what kind of allele is it? Is it X-linked? Is it autosomal? Is it recessive? Is it dominant? Is it one of those weird diseases on the Y chromosome? we can use this information to find this out. So very cool tool that we use in genetics. Hopefully you learned a lot from this. Also, I hope you got a pretty good understanding of how we can apply the product rule and the sum rules to these types of things to help predict the probability of inheriting things. So very cool application of some other stuff that we've learned through my videos. So thanks for your attention. Hopefully it was understandable, and I will see you all real soon.